Hope everyone is doing safe and well during these pandemic times. I, Sanjay from Grade 10 of Rajesh International, is honored to introduce the highly respected Madam Sarah Robertson, a Fellow of the Royal Society of Chemistry UK. A word about the Royal Society of Chemistry UK. The Royal Society of Chemistry supports professionals in the chemical sciences, sets standards for the profession, and works with teachers to inspire the next generation of chemists with the goal of advancing the chemical science. It was first formed in 1980 with the new Royal Charter and the dual role of the Learned Society and the professional body. The society had combined membership of over 35,000 in the UK and a further 8,000 abroad. The headquarters of the society are in London. It also has offices in Thomas Brown House in Cambridge, UK. The organization carries out research, publishes journals, and books and databases, as well as hosting conferences, seminars, and workshops. It is a professional body of chemistry in the UK. The designation FRSC is given to a group of elected fellows of the society who have made major contribution to chemistry and other emphasis disciplines such as biological chemistry. Madam Sarah Robertson has the designation of FRSC for her contribution to chemistry. She was the head of publishing Nelson Pounds for over half a decade, editorial director and school director of the Oxford University Press for over 7 years, and present she is the Director, Education and Professional Practice at the Royal Society of Chemistry UK for over half a decade. It's a great opportunity for us to have such an eminent person from the Royal Society of Chemistry UK. I welcome Madam Sarah Robertson for her valuable experience and to share some wisdom with us today.
Hello everyone, my name is Sarah Robertson and I'm Director of Education and Professional Practice with the Royal Society of Chemistry, or the RSC. The RSC is the professional body for chemical scientists working all around the world in all sectors of chemistry. And we provide them with support, with training, we are a voice for chemistry, and we enable chemists to connect and discuss with each other the challenges that they're solving in their chemistry careers and roles. So we're very passionate about chemistry, as you would expect. And as part of that passion, we're also very committed to and passionate about chemistry education. It's incredibly important to us that students have a good education in chemistry, because students today are the chemical scientists of the future, and it's you who are going to be using chemistry and applying chemical science knowledge and expertise and skills in solving some of the world's problems today. So what is chemistry and why do we care so much about it at the RSC? Well, chemistry is all around you. It's the air that you breathe, it's the food you eat, it's the clothes that you're wearing. All of those are chemistry. Chemistry is the study of substances, what they're made of, how they interact with each other, and the role that they play in living things and in the world around you. Chemistry is applied in research from outer space right through to the depth of the oceans on Earth and everywhere in between. And studying chemistry helps you understand the world around you. It also forms the basis of a wide variety of related subjects, such as environmental chemistry. It's so important to the world, understanding and solving challenges such as climate change, pollution, waste management, and all of those at a molecular level. Materials chemistry is the study of chemical structure of materials, and scientists who work in this field are developing for us the materials of the future. Chemical engineering is the study of how to produce chemical products at an industrial scale, again, incredibly important for the world now and in the future. And chemistry also underpins a large number of areas within the biological sciences. So biochemistry, molecular biology, and pharmacology are all underpinned by chemical science knowledge. So chemistry is used to make a difference to the world. Chemists and chemical scientists are the people who will be fixing the future for the world. Breakthroughs in chemistry impact our everyday lives, and chemists are playing an important role now in shaping the world around us, in solving the big problems, and in changing people's lives through new medicines and materials, as well as fixing the future with new energy sources. So if you start with chemistry, and by studying chemistry, you can end up changing the world. But students study chemistry for many other reasons as well. Studying chemistry helps you to think analytically and logically. It gives you skills that many employers are looking for in a wide variety of careers, not only in chemistry itself. It gives you a really good foundation for a large number of different jobs. It's a core subject, so it enables you to cross over into other core sciences or venture into areas such as biochemistry, geochemistry, chemical engineering, or physical chemistry. And it can lead to a wide variety of careers. And many people like us simply love chemistry and want to study it for that reason. So whatever your plans for the future, having a chemistry qualification could really help you. It will increase your scientific knowledge. It will help you understand why and how things happen. And it will give you practical hands-on experience. And you'll also gain important skills that are sought after by all kinds of employers. In the future, people will be needed who are good at explaining how things work and solving the chemical, biological, and engineering challenges in these sectors. There'll be a demand for people who can design and develop products for a better future. And a qualification in chemistry gives you these skills and will be a valuable asset to you if you choose to go into that area. So I hope I've been able to share a bit of information about why I'm so passionate about chemistry and why I do my job here with the RSC. You can find lots more information about jobs in chemistry and other information about chemistry itself on the website, on our website, the RSC, that's rsc.org, and also our website specifically focused on careers in chemistry called A Future in Chemistry. I hope that some of you will decide to take your chemistry study further, and maybe some of you will decide to go on to be chemical scientists in the future. We really need you. The world needs you to be chemical scientists. And I hope that if you do, some of you will join us as members of the Royal Society of Chemistry and our community of chemical scientists around the world. Thank you.
On behalf of our chairman, Dr. S.A. Joyraja, our CEO, Mr. Kuriakos M.O., our principal, Mr. Johnson Varghese, and from all the teachers and students, we thank you, ma'am, for your effective experience sharing and the link provided for us. Thank you.
A pleasant good morning to one and all. My name is Aksa Ashish from grade 9B, and here comes Ignite 2021 Science Projects Part 2.
Good morning to all. I am Asatitis from grade 7B. Today I am going to show you how to isolate DNA from a banana. Let us first make our precipitating agent. You'll need a table of salt, some hand wash detergent, and a little bit of hot water. Let us add them all into this cup. And we'll add some water. Let us give it a mix. Next we need is some ripe bananas. Let us now smash them. Here we have our banana mix puree, and here we have our precipitating agent. Now let us mix them both together. Here we have the combination of the precipitating agent and the banana puree. Now let's filter it. This is the mixture we got after filtering the precipitating agent and the banana mixture. Here I have poured the filtered mixture into a glass. Now I'm going to add isopropyl alcohol into it. Here we can see a mixture being separated. The white fibrous mass that is being formed is the banana's DNA. The white fibrous mass that is being formed is separated by a clear liquid on the top. The precipitating agent kills the banana cells and dissolves into the water. When I added the alcohol, it precipitated as a white fiber.
Good morning. I'm Dynamic Thomas of class 10A. Today, as part of my chemistry project, I'm here in the model presenting the allotments of carbon. As you can see from the model, there are mainly 3 allotropes for carbon. Those are diamond, graphite and fullerene. Diamond is made up of 3-dimensional molecules. Each carbon atom in a diamond is covalently bonded to 4 other carbon atoms in a tetrahedron. It is a well-known allotrope of carbon that exhibits hardness and high dispersion of light. It is the hardest known natural mineral and finds applications in cutting, drilling, jewelry and as well as potential semiconductor material. Graphite is not a transparent material. It is black, shiny and opaque. It is also a very slippery one. It is used in pencil lids as the layers easily slide onto the paper, leaving a black mark. It is a component of many lubricants such as bicycle chain oil. It is insoluble in water, has a high melting point, and is a good conductor of electricity, which makes it a suitable material for electrodes needed in electrolysis. Fullerene. Fullerene is an allotrope of carbon whose molecule consists of carbon atoms connected by single and double bonds with fused rings of 5 to 7 atoms. They have weak intermolecular force between the molecules. It has a hollow, ball-like or cage-like structure and has a very high melting point. That's all about my model of allotropes of carbon. Thank you.
Good morning to all. My name is Aksa Ashish and I'm from grade 9B, and today my project is about osmosis. Before we get into it, what is osmosis? Osmosis is the movement of a solvent such as water through a semi-permeable membrane as of a living cell into a solution of higher solute concentration that tends to equalize the concentrations of solute on the 2 sides of the membrane. As you can see, I'll put a few raisins into a glass of water. After a few hours or even a day, you can see that the size difference of the raisins. This is where osmosis takes place. Now, why does this happen? All this is due to the process of osmosis. The water molecules pass the cell membrane of the raisins and the raisins thus get so swollen. So you can see the size difference. I have a raisin that was not added into water. I'm going to be taking one raisin out of this water right now. You can see the size. You can of course see the size difference between all of this. This is the one which was kept in the hypotonic solution. This is not in anything. You can definitely see the size difference between it. That's all for today. Thank you and have a nice day.
A very warm welcome to all. I'm Shiva, and my topic for Science Week is photoacoustic imaging. These days we can only see inside our bodies like this or like this. But the emerging modern technology has got way more different perspective. The problem with today's body imaging technology is that the imaging depth is limited. Seeing a 3D image of inside a body is possible by a method called photoacoustic imaging. Photoacoustic effect was discovered by Alexander Graham Bell. Promising procedure does not involve any type of danger or radiation involved. See, the hemoglobin in the blood and the calcium in the bones are chemically different from each other, or simply they have different chemical structures. The photoacoustic imaging takes advantage of these chemical differences by sending pulsed laser light to a body. So this specific body part absorbs the light and then heats up. Rise in temperature makes the tissue expand and cool at the same time. So this forms a cycle. It will be like an oscillating movement. Now it's when the oscillating pressure is produced. This oscillating pressure will be similar to a sound wave. That's why it's called a photoacoustic imaging. Photo means light. In this case, it's the pulsed laser light. And acoustic means sound. And it's the oscillating pressure. Ultrasonic detectors catches the sound waves from the tissue and sends it to AI. And that formulates and creates a 3D image of the tissue. Yes, we do have imaging technologies that has damaging radiation that penetrates into a body, and exposing someone to this radiation can be bad. An interesting fact about this discovery is that it is a cross-disciplinary invention of IT and bio. If we keep advancing like this, we can shrink the world into a better one. Thank you.
I'm Alan from class 7. I'm going to explain water pressure. Water pressure is a force created by the weight of water. Now let's see an activity that shows water pressure. Fill a bottle full of water and simply poke some holes at the different heights. We can observe that the stream coming from the topmost hole lands near the bottle and the stream coming from the bottommost hole lands farthest. This is due to water pressure. Thank you.
Good morning. So today what I'm going to be doing is an activity to show the effect of detergent powder on a stained cloth. So here I have two glasses of the same volume, detergent powder, bottle of water and tea stain, which I'm going to stain the cloth with. So here I have stained 3 cloths with the tea stain. Just going to put one of the cloth in one of the glasses and another cloth in another glass. Okay. Now in one of the glasses I'm going to add, in both glasses I'm going to add water, making it almost the same volume. Yeah, they're almost the same volume. Next I'll be adding 2 spoons of detergent, that is washing powder, in this glass. And now I'm going to stir both of them for 10 seconds, and after 5 minutes I'm going to stir them again for 10 minutes, like so. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now I have stirred the cloth inside the detergent solution. Now I have cleaned the spoon because I've added it in here. So I'm going to stir this as well. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And I'm going to be moving this cloth onto a clean plate. Now after 5 minutes I'm going to stir both of the glasses again for 10 seconds. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so I've cleaned up the stirrer, and now I'm going to stir this glass as well. Yeah. 2, 3, 4, 5, 6, 7, 8, 9, and 10. This is after 10 minutes. I'm stirring it again for another 10 seconds. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. The other one as well after cleaning the stirrer. Okay, now this glass. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, this is after 15 minutes. I'm going to stir this one last time, and I'm going to keep it on the plate. I'm going to stir this now. Okay, so I'm going to take it out. This is just after the stirring process. Yeah. Okay, so so this is the cloth which was put in the detergent solution. This was the one which was put in normal water, and this was the one which was just untouched. It was put in the plate. So as we can see, the one which is in the detergent is much more whiter than the others. This has just changed only a little bit, whereas the stained one is stained. So this is the effect of detergent powder on stained cloth. So this concludes the activity. Thank you.
Good morning to one and all present here. Today I'll be presenting you a model of the stomata. But before I present you the model, I'll be sharing a few information about a stomata. So there are small openings present on the lower side of the leaves, and these pores are known as stomata. The stomata basically has four main functions. The first function is that it helps in transpiration. That is, it helps in the loss of excess water from the plants. And the second function is that while transpiration, it creates an upward pull inside the plants, and this pull helps in the absorption of water from the roots. And the third function is that it helps in gaseous exchange. And the fourth and the last function is that stomata allows the exit of water in the form of water vapor, which helps in cooling. So as you can see, this is the model of the stomata. So here first you can see that this is the nucleus, and the blue area right here, that is the vacuole, and these green dots, that represents the chloroplast, and the area right here, that is the cytoplasm, and the whole borderline, that is known as the guard cell, and this area, this particular area, that is known as the cell wall. And this is the stomatal pore, and this lining around it that is known as the inner thick wall.
Hi, I'm Sandra Marie Jones from grade 7B. Ecosystem. Ecosystem is a community of living organisms along with their natural habitat and the non-living components of their environment. This network of non-living and living components is a given area is known as an ecosystem. An ecosystem could be a pond or neither a house. Also, some people believe that our earth is an ecosystem too. Ecosystem includes two major parts called biotic and abiotic. Abiotic components are the non-living elements in the system such as air, water, nutrients, sunlight, and temperature. Biotic components are all the living organisms like animals, plants, fungi, microorganisms, and you. And thank you.
If you want to know how a plant grows, it takes water, air, sunlight, and makes cellulose. Every plant can do this fundamental process, and we can call this photosynthesis. Unlike me and you, plants use CO2.
Today I have made a mini robot. So this is my mini robot. I am going to turn it on. You can see that it's moving. It is moving because of the air resistance. This is my mini robot. You can see. There is air resistance. That is why it is moving.
I am Emil Kottayam from grade 10A. Structure of diamond. Diamond has a gain covalent structure in which each carbon atom is joined atom by 4 outer carbon atom is covalently bonded. The carbon atom have a regular lattice arrangement. There are no free electrons.
My dear teachers and my dear friends, today I am going to explain about cells. These are the few examples of cells. This is plant cell, this is root hair cell, this is animal cell, this is human cell. We can't see these cells with our eyes. We have to use a microscope to see these things. All living things has a cell, but all non-living things does not have a cell. These are the starting point of a human body. Thank you.
Hi, I am Anai Bobby and I am going to do a presentation on artificial intelligence. Artificial intelligence AI refers to the simulation of human intelligence in machines that are programmed to think like humans and mimic their actions. The term may also be applied to any machine that exhibits traits associated with the human mind, such as learning and problem solving. There are 4 types of AI: reactive machines, limited memory, theory of mind, and self-aware. What is the purpose of AI? The basic objective of AI, also called heuristic programming machine intelligence or the simulation of cognitive behavior, is to enable computers to perform such intellectual tasks as decision making, problem solving, perception, understanding human communications in any language, and translate among them. Why artificial intelligence AI is becoming important? AI technology is important because it enables human capabilities, understanding, reasoning, planning, communication, and perception to be undertaken by software increasingly effectively, efficiently, and at low cost. Use cases are proliferating as AI's potential is understood. What are the positive effects of AI? Artificial intelligence can dramatically improve the efficiencies of our workplaces and augment the work humans can do. When AI takes over repetitive or dangerous tasks, it frees up the human workforce to do work. They are better equipped for tasks that involve creativity and empathy, among others. What are the advantages of AI in education? With the use of AI, students have a personalized approach to learning programmed based on their own unique experiences and preferences. AI can adapt to each student's level of knowledge, speed of learning, and desired goals so they are getting the most of their education. What are the disadvantages of AI in education? Lack of personal connections. While smart machines improve the education experience, they should not be considered as substitute for personal interaction. Relying too much on these machines to grade our tutor may lead to educational oversight that hurt learners more than help. Thank you.
Good morning everyone. Today I'm trying to explore the study of plant population frequency using the quadrant method. Our aim is to study the plant population, that is the frequency of an individual plant in a particular area. This is a simple quadrant which I have made. Normally these quadrants range from different sizes to shapes. I am placing this quadrant in a particular area to check the population of this plant, which is known as little tree plant. So let's get started. Even though this is a simple method, it is objective. Frequency is highly influenced by the size and shape of a quadrant. So the habitat we choose must not be too big or too small. Let us look at the frequency of these plants. So here we need to check where all the little tree plant is present, as you all might know, which I had shown before. Here we found one, so we place a small stick there to identify the place. Second, where do we find that? Yes, over here. And third, over here. So let's mark it. Now we have placed 3 sticks in the frequency areas of the plant. Now we need to mark the population into a frequency table. I have placed the quadrant in another area. Now let's check out the frequency of the little tree plant in this particular area. We spot one plant over here, so let's mark this point. We find another plant over here, so let's mark this point too. Another one and another. I have marked my results onto a frequency table. Now let's check out the results. The first area where I placed the quadrant, the frequency of little tree plant was 3, and the second area where I placed the quadrant, the little tree plant frequency was 4. The biodiversity shows that the first area where I placed the quadrant, the biodiversity in that area is more. And the second area where I placed the quadrant, the biodiversity is less. So thus, my finding is that we can use this method to find the biodiversity and the frequency of an individual plant. Thank you.
Hello everyone, today we'll be doing a simple experiment by using a leaf. Now we all see this in our daily life, in the backyard, in the garden, in every forest, every jungle. You always see this leaf hanging around in many plants and trees. Now, as simple as this leaf looks, it is having a complex structure in molecular terms, and it has many functions, one of which is producing food by photosynthesis. Now how it does that is, if you see this leaf, it's green in color. Most leaves will come in different colors, but the most common one is green because of a pigment known as chlorophyll, which is present in the leaf, and its role is to capture the sunlight and use that light energy for photosynthesis to produce food. Now this experiment is not going to do that. This experiment is going to show you how oxygen leaves the leaf. And so, without further ado, let's start the experiment. But before we start the experiment, there should be always some precautions. Now, precautions are that we're going to use hot water for this, so please be careful while handling the hot water, because you might get burned from it. You might slip and fall if it gets dropped to the ground. And for plucking the leaf, you can use your hands, that's not an issue. But if you're using knives or scissors, you have to use adult supervision for that. So let's get into the experiment. So right now what I have with me is a bowl of hot water. And yes, it is hot. You shouldn't boil the water, but you can make it hot. So for this, first we're going to take this leaf and then dip it in the hot water. It's that simple. And what should we be expecting is bubbles forming at the underside of the leaf, because the underside of the leaf has stomata, and stomata is responsible for letting air in and out. And in this case, it's responsible for letting oxygen go out. So let's see what happens when we dip it in the water. Okay. There's no bubbles formed. Well, I guess this has been an unexpected one, but the bubbles haven't been formed on this. The main reason is because either maybe the water might not be hot enough, or it could be the fact that this leaf has been taken during nighttime. Actually, you can only use leaf at daytime, because at night there's no sunlight at all, and so it won't form bubbles because there's no oxygen releasing out of the leaf. So I think the reason could be that maybe there's no sunlight. But if you're using a fresh leaf that has been outside in the sun for a while, it will produce bubbles on the underside. Now I'll show you a picture to explain how the oxygen leaves the leaf. So this is a cross-section diagram of a leaf in microscopic levels. Now as you can see, this is the cuticle, this is the palisade mesophyll layer, this is the spongy mesophyll layer, and these two are called the stomata. Now how oxygen leaves the leaf is from this layer, the palisade mesophyll layer. Now it doesn't leave through the cuticle. Instead, the oxygen goes down from the palisade mesophyll layer to the spongy mesophyll layer, and then eventually it will reach the stomata, where the stomata will open. Its guard cell will open, and then the oxygen can leave from there. Now here's the diagram of a stomata. Now look at this. Now this is the guard cells, and when oxygen or water leaves the stomata, the guard cells actually open to let the water and oxygen leave the leaf, and that is how oxygen leaves a leaf. Not only oxygen, carbon dioxide, water, these all components leave the leaf through the stomata.
Thank you for watching and also stay tuned for part 3. Thank you and have a nice day.